Good morning, everybody. It's for me a great pleasure now to introduce my friend Dr. Lluis Montoliu, he has accepted one more year to give this lecture for the master students, not only for our master's degree, but for other masters. It's actually this talk is open to everybody. And let me start with a short presentation of Dr. Lluis Montoliu. First of all, I would say that now let's share. Okay, so Dr. Lluis Montoliu is a biologist. Actually, we studied together at the University of Barcelona. He has been deputy director of the CNB, director of VHIR, the VHIR of rare disease, and also is director of the Spanish node of the EMMA, honorary professor at the University at the Complutense de Madrid. And also he has generated a lot of animal models for human rare disease, especially in albinism. I'm not going to give more spoilers about his work. Also, he's president of the CSIC Ethics Committee and a member of the ethic panel in the ERC in Brussels. He has a very important age index and more than 200 publications. But he's also a great communicator. I mean, I especially recommend you to listen this last talk that he gave in Naucas in Bilbao. It's really special. Also, he's a mass communicator. He has an awarded researcher. He has received many different awards. The last one, the Coste of Difusión de la Ciencia in 2022. And he also is leading this network of transparency in animal research. And finally, he's a successful writer. I recommend you all these 3 books that are really interesting. And also, he's an opinion leader, an influencer with today, you have a right to almost 35,000 followers. And also, the most important thing, he's a good friend. But I don't want to stay more with this talk. I mean, the best thing that we can do is to listen to give you all to listen you in your presentation. So, Lluis Montoliu, thank you again for being here, and sorry for the problems that we have had with the link and the communication. And please go ahead when you.
Don't worry, Josep, and very good morning, everyone. Thank you very much, Josep, for this very kind introduction. So let's start right away with the presentation. Please confirm that you're seeing the first slide.
Yeah, correct.
All right. So basically today, in the next 2 hours, I'll try to finish before the 2 hours so we have some time for questions. But as I said in the chat, please don't worry, there are many slides, but I will be sharing the slides afterwards with Josep, and Josep will be distributing the slides to whoever is interested. So my task, my role today, as I do every year, is updating where we are regarding genome editing biomedicine, because this is a moving target. So every year we know more, and every year there are new applications, and every year we are facing different challenges. Actually, recently I started to prepare my talks with something which you might think it is unexpected. So this is an image that you probably don't expect in a genome editing talk, but this is an image that illustrates that we're living in a world that is fascinating. So we are capable of sending a rover to Mars, and this rover is traveling and is moving in Mars, in the Mars surface, together with an helicopter. And so this is fascinating to me, that the mankind has been able to send such a device, such a rover and such an helicopter to Mars. You probably know the names of these 2 robots. The rover is called Perseverantia, and the helicopter is called Ingenuity, Ingenio. And these are two features that I consider they are really remarkable and important for science. And as much as this Mars and helicopter, this rover and helicopter on Mars are fascinating, what I think is also fascinating is that we have been able to cure Alisa. Alisa had lymphoblastic leukemia. This is acute and T cells. And she was condemned to death because she had no medical treatment available to her. And last May, well, May 2022, she was given an experimental treatment using base editors. May 22. The base editors, as you will see, were discovered and they were presented to the community in 2016. So 6 years later, we have a technology that didn't exist at all that is now already in the clinics and it's already saving lives. So this is as fascinating as having rovers and chopters on Mars. But let's start from the very beginning. If you're landing from nowhere, if you're landing from Jupiter and you know nothing about CRISPR, what I will first recommend is to have a look to this nice review by Jennifer Doudna and a collaborator. And this review was published at the beginning of this year. And it's probably a good way of revisiting the last 10 years. So, as you know, the CRISPR technology was presented in a science paper by Doudna and Charpentier in June 2012, but it was not only 6 months later, in January 2013, when the first experiment successful demonstrated that these CRISPR tools were capable of editing genes at will. So this is the 10 years that Jennifer is referring about, and she's also contemplating the different applications, the pitfalls, the problems that they have to be solved, and the different uses that this innovative technology. When I saw this, I was focusing that, of course, we need to give credit to these two researchers, Emmanuel Charpentier and Jennifer Doudna, Charpentier being the director of a Max Planck Institute in Berlin, and Jennifer Doudna at University of California, Berkeley. They, as you know, they received the Nobel Prize in Chemistry in October 2020, so it's 3 years ago, and this was a very well-deserved prize. But when I was reading about these 10 years, I was lacking 20 more years. And actually I was lacking 20 and 30 more years. And I was lacking this because we shouldn't forget that the whole history started with this friend of mine, which is Francis Mojica, Francisco Juan Martinez Mojica. This is a macrobiologist from the University of Alicante. And these are the milestones in the CRISPR history you should know. So what Jennifer Doudna is referring in her review are the two last blue dots. So 2013, the first papers by Feng Zhang and George Church published in Science, and 2023, that's where we are now. But 10 years before, in 2003, this is when Francis Mojica had the eureka moment. This is when he realized that these CRISPR repeated sequences were the basis of an immune system that probably was used by bacteria and archaeas to defend themselves from the infections by bacteriophages. It took him almost more than 2 years to publish, and this is why the most famous paper by Francis was eventually published in 2005. Same has to be said regarding the 2013 milestone. As I said before, the science paper by Charpentier and Doudna was published in June 2012. So this is what makes 10 and 20 years. And why 30 years as well? Because in 1993, that was the first paper published by Francis Mojica, in which he was presenting, and he was first sharing that in an archaea, Allopherax mediterraneae, that he found in the salt ponds near Alicante in Santa Pola, he was able to find these CRISPR systems. These CRISPR systems, he was not the first to locate. There were other two teams, and the most important in 1987, that was the Japanese team that was seeing these repeated sequences in Escherichia coli. So, but this is in brief, what is the milestones of the CRISPR technology. And this is why beyond these 10 years, we have to refer to the 20 and to the 30 years. So these are the 20 years that go between 1993 and 2013. The first two rows, they refer to microbiologists and biochemists, and they are the ones that contributed describing the different compounds of the CRISPR technology. And the last row, probably the most relevant, are the ones that really put these compounds to work. And Feng Zhang and Church in January 2013 were the first ones demonstrating that these gene editing tools were useful and were working in mammalian cells, both mouse and human cells. Rudy Jenich was demonstrating this was also working in mice. That was May 2013. And Keith Young and others in April 2013 demonstrated this was also working in zebrafish. Usually, these are my 12 people in this Hall of Fame of the CRISPR technology, but lately, I have to add a 13th person. I have to add another person. And this is the person that has really boosted this field. And this is David Liu. David Liu is working at the Broad Institute of the MIT in Boston in the USA. That's the same institute where Feng Zhang is also working. And he appeared in 2016 with the second generation of the genome editing tools, these base editors. And in 2019, he was presenting yet the third generation of CRISPR tools, which are the prime editors. And as you will see, I'm making a small spoiler. We all thought that the first generation CRISPR tools were the ones that will be arriving to the clinic. And in some cases, this might be true. But in the long run, in reality, the ones that will be reaching the clinic, the ones that will be really used in hospitals, will be probably second, and eventually in the future, in the near future, the third generation CRISPR tools. So one remarkable feature of all these different researchers that I think must be underlined, because without this, it is difficult to understand how this field has been progressing so rapidly, is that all these basic researchers, they have a second life, they have a second personality in the industry, and they have created companies, and these companies have fueled and have really triggered the development of these applications in the hospital, in the clinics. For instance, Zhang, Church, Young, and Liu, they were the ones founding, together with other researchers, Editas Medicine, and you will see Editas is appearing in some of the clinical trials I will be commenting. Feng Zhang as well is founding other companies. Two of them are Sherlock and Arbor, that are related with diagnostics, genetic diagnostics. I will not be referring today to genetic diagnostic, but Feng Zhang played an important role in the COVID-19 pandemic, because he was the first one already in March 2020, at the beginning of the pandemic, developing an alternative diagnostic tool using CRISPR compounds that was not using PCR. And that was approved for diagnostics in the US. Unfortunately, it was not approved in the European Union, but this is to say that CRISPR, beyond being able to modify genes at will for genetic modification of organisms, including human beings, CRISPR tools also can be used for diagnostics. Jennifer Doudna has been behind the creation of several companies, the most important one being Caribou. And Caribou, you will see that is also part of some of the developments I will be referring, but also Mammoth and Intellia. Intellia is also part of some of the latest clinical trials, and Mammoth is also focused in diagnostics. Emmanuel Charpentier is probably the most successful scientist regarding the industrial applications. She was founding CRISPR Therapeutics, which is a company that is putting these CRISPR developments into the clinic. And probably she will be the first company having a product authorized for clinical treatment in Europe and in the US by the European Medicament Agency and by the FDA. CRISPR Therapeutics has another division, which is Genomics, that takes care of all the genome editing applications that have not to do with human beings. Also, David Liu, Yang, and Zhang, they created Beam Therapeutics to promote the development of the base editors. And David Liu also created a third company on the third generation CRISPR tools. This is the prime editors. And as you see the logo, as it is being progressing, they are so talented that they produce logos that they correct themselves. And this is just amazing, these guys. Okay, so basically this technology that we have inherited and we have imported from the prokaryotic world, in the prokaryotes, it has a function, it has a defensive function. This is an acquired immunity. The more viruses a bacteria is exposed to, the more of these elements in the CRISPR array that are being included in the bacterial genome. And this bacteria is resistant to the future infection of the same viruses. And this is due to an interference that is mediated by an RNA that triggers a cascade of nucleases that eventually kill and destroy the DNA of the intruder. So this is a very effective and evolved system to defend these prokaryotic cells from bacteriophages and from other plasmids. We can transform and we can transport all these different elements, say at the DNA level or at the RNA level or at the protein level to eukaryotic cells. And there we can promote the additions. You can have a look at in this review that we published a couple of years ago, Francis and myself, where we described the basis of this technology. Basically, we're dealing with nucleases. So the first generation CRISPR tools are cutting DNA. And because they are cutting DNA, so they are triggering this repair response. So probably this double-strand DNA cut is one of the most aggressive insults you can do to a cell. Cell being a prokaryotic cell, cell being a plant cell, a G cell, or a human cell, or an animal cell. And this double-strand break, they needs to be repaired immediately because otherwise they will be a part of the chromosome that will be lost in the next division, because this part of the chromosome that is not attached to a centromere will be lost when the cell will be dividing. So we have, at least we have two systems. We have several ones, but we can summarize in two systems. On your left, we have the non-homologous enjoining, which is a system that is equipped in all our cells. So all our cells are, they can process these double-strand breaks through this non-homologous enjoining, which is inserting and deleting nucleotides randomly. And inserting and deleting, this is what we refer to indels. And these indels eventually they create a microhomology that is used to seal, to match these two different strands, and eventually the ligases, they do their job, and eventually the whole chromosome is physically reconstituted. Of course, because we have inserted and deleted nucleotides, what happens is that the gene is probably disrupted. And this, which at the beginning it could be considered as a down effect, as a negative effect, in reality, it's an advantage, because this is one of the best ways that we know today of inactivating a gene. So basically, we use a CRISPR that is targeting our favorite gene, and through the use of these first generation CRISPR tools, the result is that the gene is no longer working. So there is another system, which is the one on your right, that is the homology-driven repair system, in which we can provide an additional sequence that shares homology left and right to the double-strand break and provides de novo sequences. And this is properly called the gene addition. And we all thought, including myself, and if you revisit seminars or webinars that I was giving in 2013, 14, 15, etc., at the beginning of these 10 last years, we all thought that the homology-driven repair would be our holy grail. So this will be the solution to all of our problems, and this will be the approach that eventually will be reached in the clinic. So we were all wrong. So what happens is that we know nothing or almost nothing about the homology-driven repair proteins, and we know that it's only operating in a few cells, in a subset of cells. In principle, in dividing cells and in some of the dividing cells. So, for instance, if we have to modify a gene that is expressed in neurons, and neurons, most of them, they do not divide at all in the adulthood, basically, we're done. We cannot use homology-driven repair. We have to use non-homologous enjoining. So that was a first limitation that we learned the hard way. So after trying and after realizing that academically, in my lab, we do HDR all the time with mice, but I can handle with a success of a 2%, of a 1%, or 5%, because I select the mouse that carries my desired gene mutation and I kill the rest. I cannot do this with patients. I cannot go with a 5% efficiency to the clinic. I cannot afford just giving this to 20 patients and just hoping that one will be eventually corrected. So this is why we had to go back to the non-homologous, or we had to develop further systems, which is what actually happened. So this is what was presented in 2012 by Jennifer Down and Emmanuel Charpentier. This is a binary system. Originally, it's a synthetic guide RNA that has a common part at the end, at the 3' and at the 5' has 20 ribonucleotides that are pairing with the corresponding sequence complementary in your favorite gene. So this is triggering a double-strand break. These are these small red triangles. And this is only happening when the RNA guide is next to a sequence. And this is the protospace adjacent motif, the PAM. And this is a biological requisite. And this is just to remind us that we're using a biological tool. This is the trick that the bacteria is using not to kill itself. Because without the PAM, the bacteria will not know whether this sequence that is inserted in the genome is the one that the bacteria has to destroy instead of the DNA from the intruding bacteriophage. And the difference being that in the bacteriophage genome, there is a sequence next to the guide that this is what the nuclease is looking for. And when it locates this PAM sequence, then the nuclease, they know that they are acting on the bacteriophage genome and they are not acting in the bacterial genome, because the bacterial genome only has the complementary sequence and it doesn't have the PAM. So this is the reason why we have to locate our guides next to PAMs that are different. They can be, as in this one, at the 3', they can also be at 5', they can be different sequence depending on the nucleases. And also, we have now nucleases that have been evolved in the lab, and they are recombinant, they are mutated, that they are operate in a PAM-less fashion. So now there are nucleases available that they don't need PAM anymore. But that was the origin. So 11 years later, what we have in at our disposal, so what we can buy, is a three-part compound. This is the first generation tool, and this is what we call ribonucleoprotein. So all what you see in the slide is commercially available. And this is important because this explains why this has been evolving so rapidly. So we have the Cas9 nuclease, or any other nuclease you might think of. Then the guide has been split into the two original compounds, which is the CRISPR RNA, which is the variable part, which is the one that targets your favorite gene, and the tracer RNA, which is the common part that pairs to the CRISPR RNA and hooks to the nuclease. So these are the three compounds that you have to design when you are preparing a CRISPR experiment. Essentially, you buy Cas9 and tracer RNA, and they are valid for all the experiments. And the only thing that you have to change from experiment to experiment is the CRISPR RNA. So that's pretty easy, and this is working nowadays, so that you can apply this to a number of different applications and to a number of different solutions. And this is what I will be describing next. So the easiest application of the CRISPR technology is for disrupting a gene. And this is what has been used mostly in most organisms, not only in human beings and in human cells and in mouse cells and in mice, but also in plants. In plants, the first use is also disrupting a gene. And why is that? Because that's the easiest. Basically, the only thing you have to prepare is a CRISPR complex that is targeting a gene of your interest, and then upon this gene being cut, and upon the cell being repairing this double-strand break, in most of the cases, the gene will stop working, and you will be generating a cow. You will be generating a knockout. Where this can be important, well, in different fields, but in biomedicine, I want to highlight the use in something that has been appearing in the literature, in the scientific literature, that was forgotten for many years. So xenotransplantation was an idea that was appearing in the 90s, in which you can genetically modify pigs so that their organs, they can be used for transplanting into human beings or into primates, put it this way. However, you have to modify some genes so that the immune system is not rejecting these big organs, because otherwise they will be immediately rejected. Actually, the pigs that are now being prepared, they have at least 10 genetic modifications. And this is probably the most heavily genetically modified animal, or I would say animal or plant or yeast or bacteria that is existing. I know no other animal that has so many genetic modifications. In particular, these pigs, they have 4 genes inactivated, and guess which is the system they're using for inactivating these genes. So they're mostly using CRISPR, and they're also adding 6 human genes from the complement reactions to modulate the immune response. So these are the genes that are normally being used, and I draw your attention to one of the genes that is inactivated is the growth hormone receptor. And why is this? Well, they're not using standard pigs, they're using mini pigs. Well, mini pigs means is a pig that has 80 kilos. That's similar to a human being. And mini pig, still, when they are using their heart from young mini pigs, this heart, when it's being transplanted into the thorax of a macaque or of a baboon, or eventually to the thorax of a human being, thing of a child, then this heart will continue growing. And this is a problem, because sometimes it grows so much that it cannot beat anymore inside the thorax. So you have to prevent this from happening. And one way of preventing the continuous growth is the inactivation of the growth hormone receptor, which is not a gene that people might think of that will be relevant for xenotransplantation. So this is the pathway you have to follow for xenotransplants. And for more than 30 years, the pathway was stopped at the baboon. So we knew how to produce these somatic cell nuclear transfer mediated pigs. We could test it in vitro, we could test it in non-human primates, but we had no experience in transplanting these organs into humans. This thing changed with the participation of these people from New York, from the Transplant Institute in New York, Robert Montgomery and colleagues. And the first xenotransplant that was reported was in September 2021, in which in a woman that was clinically dead. And so she was ventilated and she had a heart beating, but it was with no brain activity. She was xenotransplanted a kidney from one of these pigs, and she was they attached this kidney to one of her legs. And the reason is that they wanted to see whether the rejection was occurring or not. and they connected to the blood circulation, and they could see for more than 50 hours that the kidney was not rejected and the kidney was functioning and producing urine. This has been now repeated in this in different experiments, not only with pigs but with hearts. In all these cases, they were transplanted to cadavers. They were transplanted to dead people, to clinically dead people, for testing the rejections. The first proper xenotransplant was occurring in January 2022, last year, and it was holding for 2 months. In March 2022, David Bennett, this patient, he was needing a heart transplant, but he had so many other comorbidities that he was not a very good candidate for transplantation. So he was about expecting to die, and the FDA, he was authorizing a compassionate use, and this compassionate use was providing this first opportunity to testing one of these pig hearts into a human being. Eventually, this person did not die of the consequences of the rejection, but he was dying because he was not only xenotransplanted, but he was also put under severe immunosuppression regime, of course, because they need immunosuppressors. And what they didn't control or they didn't expect is, together with the heart of the pig, they also transmitted these porcine cytomegaloviruses. And these cytomegaloviruses were the ones that eventually killed David Bennett 2 months. So this is a warning that tells us that it's not only that we have to care about the immune system rejecting the organ, but also we have to care about the health status of the pigs that are going to be used. So there is a second heart xenotransplant in its way. So this has been commented in September, so last month, and we know nothing. So we know that this is ongoing, and apparently everything went well. This is another person. So I wanted to tell you that these pigs that we are using, they somehow are related to Dolly the sheep, the first animal clone from adult cells. Because Dolly, as you might remember, was produced by a company in Roslin, PPL Therapeutics, that eventually went bankrupt and was purchased by Revivicor in 2003, that eventually also went bankrupt and was purchased by United Therapeutics in 2011. And in the last years, in the last 10 or 12 years, United Therapeutics has been creating these pigs, that these are the ones, thanks to the CRISPR technology, that are reaching already the clinic. And this is not only the 10 genes, there are almost more than 20 genes that can be inactivated or overexpressed for the success of this CRISPR technology and for the success of this xenotransplantation. And CRISPR technology is the application that is making this possible. You probably heard a couple of weeks ago that there is another strain of pigs that is being presented for xenotransplantations. And this is coming from George Church in Harvard. George Church in 2017, he was using CRISPR to inactivate the 69 insertions of the porcine endogenous retroviruses, which is one of the dangers of these xenotransplantations. In 2017, they created these pigs, and it took them a while to add additional genetic modifications. And they just published 2 weeks ago that kidneys from these pigs, they could survive into macaques for more than 2 years. So this is already another series of pigs that could be used eventually for xenotransplantation. But these ones, they have not been yet used in human beings. But I want to highlight this fact that with using CRISPR, they could inactivate not 1 or 2 or 3 or 10 or 20, but 69 insertions of the PERVs into the pig genome. Last but not least, on this disruption regime and disruption applications from Roslin as well, this past week, you could probably read in Nature Communications that in Roslin they have produced chicken that are resistant to influenza virus. And you know that chicken and in general birds are the ones that are transmitting this influenza and pandemic every year. So this endemic influenza that is reaching us every year is transmitted by this bird. So it is important to know how to block. And they realized that they could add by CRISPR 2 mutations in a protein that is required for the viral replications. This is AMP32A. And also they realized on chicken cells that they could inactivate 3 members of this family of NPS proteins that are involved in the replication of the virus. These are genes from the chicken cells, and upon inactivating this, the virus was not replicated anymore. So this is good news for the future. So what else we can do with CRISPR? Well, we can do deletions. And now it's obvious to all of you, because probably you all have grown and have already studied CRISPR. But when we started, and when I say we, I mean my lab, when we started in 2013 using this technology, we didn't expect this would be so successful and so robust, this technology, because we speculated that if we can target 2 sequences in cis means the same chromosome in the same DNA molecule, we speculated that the repair system will be sometimes committing a mistake and will be sort of sealing the left and right parts of the chromosome and leaving behind and deleting whatever it is in between 2 consecutive CRISPR targets. Now we know that this is very effective, and this is very frequent, and this is probably the second most frequent application of CRISPR. First one being inactivation of genes, second one being the creation of deletions. And that has an impact in our research. We are investigating rare diseases, including albinism. This is a disease that is not only impairing pigmentation, but is also essentially impairing the visual capacity. So these are legally blind, these people. There are many genes involved in albinism, and this is what we have been studying. These are the 22 genes that are resulting in 22 different types of albinism. And these are the ones that we are studying. And particularly the first one and the most common type of albinism, which is also associated with mutations in tyrosinase. Tyrosinase is an enzyme that is at the basis of the biosynthesis of melanin. Obviously, if you have tyrosinase mutated, you are not synthesizing melanin any longer. What happens is that not only with albinism, but also with other rare diseases, about 30% of patients we fail to genetically diagnose. In the lab, thanks to our collaboration with the VHIR, Theory of Rare Disease, we are doing genetic diagnosis of many Spanish families with members with albinism. But 30% of them we fail to diagnose because mostly we are focusing into the coding part. In the non-coding part, in the intergenic part, there are regulatory elements that for many years we speculated whether they would be relevant for the expression of the gene. But these were very difficult to be targeted because they were surrounded by repeated sequences. They were surrounded by retroviruses, transposons, retrotransposons. So any attempt to go there by homologue recognition would be failing. And the solution was, when CRISPR technology was appearing, is to target 2 double-strand breaks with CRISPR left and right to your regulatory element, the one that you wanted to evaluate the function. And this worked very nicely. And we didn't know at the beginning that this would work so nicely. But the reality is that you could, using bioinformatic tools, you could locate, the only thing you need left and right is 20 unique nucleotides that identify a unique sequence in the genome so that you can build a CRISPR complex and target this sequence left and target another sequence right to the regulatory element. And if everything goes well, you will be deleting this regulatory element. So we did this, and we have published several attempts, and you can see this in the literature. This is just an example. And this is when we are deleting an element that sits 15 kb upstream 5' of the tyrosinase promoter. So you don't have to be very talented, and that's very easy to realize that the mouse that is sitting on the top is gray, which means that we are impairing the expression of the gene. Mind you that the gene is intact. We are not touching the gene itself. We are only touching this regulatory element. And this is the first time that we could confirm that you could eliminate one of the regulatory elements and cause a phenotype that is so clear. So when we saw this and we repeated this with different regulatory elements, now these regulatory elements, we are also using them, the homologous counterparts in the human genome, for exploring for the potential, for potentially finding mutations that could explain the albinism of people. So using all these different technology, we could reconstitute the tyrosinase topological domain. And this is something that it can only be done with a very few number of genes. So we know nothing about these regulatory elements for most genes. And for tyrosinase, thanks to CRISPR technology and thanks to the work we've done in the lab, we could speculate that there are 2 CTCF sites that are creating a loop that contains not only tyrosinase, but another gene that is ubiquitously expressed, NOX4. Now we have in cells that are not expressing tyrosinase, the 3' regulatory element is creating another small loop in the tyrosinase. In cells that are expressing tyrosinase, such as melanocytes in the skin and the iris and in the choroid, then the 5' element is also bound to the promoter. And in another type of cells that are expressing the tyrosinase in the retinal pigment epithelium, in the retina, in the eye, this CNS2 regulatory element is also bound to the promoter. So this is the first time we can explain how one of these mammalian genes is operating, because we have been able to dissect all these regulatory elements. So this is the power and the capacity of these CRISPR tools. Another set of experiments that we have done that I wish to comment, because I want to illustrate this also with our experiments, is the creation of point mutations. And this is something that for years it was very difficult before, but now we can use the homology-driven repair. Mind you that this is very infrequent, so it's very rare, but we don't care. Basically, we are selecting the cell, or we are selecting the mouse that carries this mutation, and this is the mouse that we're using. This is not to be transferred to the clinic because of this low probability of this low efficiency. But academically speaking, using animal models, this is a fantastic tool, and this is something that has been enabling us to create what we call the avatar CRISPR mice. Avatar, because we're using this metaphor from the 2009 movie from James Cameron with this blue beast, the ones that are linked, each one of them, to a human being. And I'm using this metaphor because basically I'm diagnosing a patient, I'm finding which gene and which mutation he or she is carrying, and exactly the same mutation using CRISPR, I'm transmitting this mutation to a mouse, to the counterpart mouse gene. And therefore, the resulting mouse that carries the same mutation of that person is the avatar of that person. And this is what we have been doing for different types of albinism. This is, again, another example of a tyrosinase mutation. This is a person we have diagnosed in the lab that carries a mutation in tyrosinase. It's the albinism oculocutaneous 1A. And this is the corresponding avatar that is being pointed. As you can see, they have the same phenotype because they have the same mutation. This is an intermediate mutation. It's called OCA1V, in which this person is a person with albinism, even though it has some pigmentation postnatally. And this is what we have recreated in this mouse that has this gray color. And then we have this albinism that is being characterized in Africa, and this is OCA2, oculocutaneous albinism type 2. Please pay attention to the color of the hair of this guy. This is a black person from Cameroon with albinism. This is why you don't see pigmentation. And this is exactly the avatar. And as you can see, the color is the same because it's carrying the same mutation. And we have done the same with this person that carries another type of albinism out of these 22. In this case, it's a deletion inside the coding part of a letter, of a nucleotide, and this is changing the open reading frame and creating a mutated protein. And this is the mouse that represents the avatar of this person. Why are we creating these avatars? Because we can study the consequences of every single mutation, and we can also use these avatar mice for validating compounds or drugs that are repositioned, which is one of the best ways of trying to find new drugs. So drugs that have been already approved for another pathology, that now we can test whether they have an a secondary use in this pathology. And this is what we have done with DOPA or nitisinone, that they were used for other pathologies. So we have done a number of avatar mice, and these are the ones that we have been created. And I want to share the results of 2 of them, because these are the ones that we are recently published. We published it just last month, and one that is almost ready for publications. So this is the first case. First case is a new type of albinism. It's called FONDA. FONDA, it's an acronym that refers to this fovea hypoplasia and optic nerve decussation effects and anterior segment deficits. This is the acronym, and it's a type of albinism that has the same visual abnormalities, but has no pigmentation defects. So the people affected, and this was described in a family by some Dutch geneticists, and there are 8 families being described. And we wanted to mimic, and we wanted to create the avatar of one in which this glutamine 200, which means at this level of the protein, we recreated a stop, and the protein was truncated. And here we are. We generated many mice, and due to the mosaicism, we generated many different alleles. And one of them was almost closely resembling to what we wanted. We wanted to have the stop at the glutamine level, but as you can see, we created the stop at the immediately before amino acid, which is the proline. And we decided to carry on with this. We confirmed that this was resulting in a truncated protein, because in homozygous CRISPR mice, the CN using an antibody against the carboxy end of the protein was not detecting anything, whereas the central antibody was still detecting the protein. So this is how these mice are looking, and this is exactly what we wanted to see, because as I told you, the corresponding families, they don't lose pigmentation, and these are the mice at the heterozygous or homozygous level that similar to the wild-type pigmented mice, they don't lose pigmentation. The question is whether they have visual abnormalities. So we focused first at the histological level, and we saw some decrease of pigmentation at the level of the retina that you could not see with the naked eye. So you have to prepare these histological sections. And when we did these electronic microscopy images, what we saw are these structures, these back walls, and these alterations in the RPE cells. What you're looking at are RPE cells, and what you're looking at the last row are the melanosomes, which are these lysosome-related organelles that are producing the melanin. So there is an impairment, there is some malfunctioning of this RPE due to these mutations in this gene, this solute carrier T8A8. So what about the connections between the eyes and the brain? If we compare wild-type pigmented and albinos, what you see, and this is what the arrows are pointing, is that the ipsilateral projections, that means the projections that comes from a retina and they go to the same side of the brain hemisphere, that they are normally there, in the albino, they are absent. And when we saw the heterozygous, we could still see this, but in the homozygous, we could not see, similar to what happens in the albino. So we reproduce this these axonal projections alterations in these mice. We also measure this with a more sophisticated technique in collaboration with Alexander Repsam, Institut de la Vision in Paris, in which we are injecting different neuronal tracers in each eye, left eye and right eye in red and green, and then we can clear the whole skull and the whole brain, and we can use a microscope, and this is light sheet microscope, to see the entire system, the entire visual system. When we quantify these connections, we see that the homozygous SLC38 CRISPR mutated animals, they have a reduction. Not only this, we also, together with electrophysiologists, with Nicolas Cuenca, we saw that there were electrophysiological alterations, and also with the optometer test, which is measuring the visual acuity, we saw difference in these mice. So basically, it tells you that we can apply CRISPR technology to investigate this new disruption. We have done the same with another type of albinism, which is the ocular albinism type 1. In this case, there's another gene involved. It's called GPR14033. And again, this is a type of albinism in which pigmentation is not affected. We had a control mouse that was a standard knockout. This is a gene that is inserted in the X chromosome. So basically, if we do the experiments in males, basically the males are knockouts in just one X, because that's the only X chromosome they have. And we created the avatars of two patients that we could diagnose. On your left and the bottom row, a P300L, so a proline change to leucine in 300 residue of the protein. And also we created a splicing variant. So often patients, they have mutations at the splicing branching points, and this is also creating alterations. And we prepare these mice, they're still pigmented. When we measure the expression of the genes, logically, the one that has only an amino acid exchange, it is more or less expressed like the wild type, whereas the one that is knockout and the one that has a splicing variant due to the disruption of the expression, you have a decrease. Also, the one that has this splicing variant, when we investigating, it is skipping one exon, which is the result, which is the cause of this alteration. We investigated against the retina in at the histological level and at the microscopy level, at the electron microscopy level, and what we could see in this ocular albinism avatar mice is the presence of these giant melanosomes. The giant melanosomes, they are characteristic of the ocular albinism. We could also see that these mutations, they're also reducing the number of fibers that are maintained in the same hemisphere. So basically, these are two examples that illustrate the power of how powerful are these techniques to create avatar animal models and to investigate them. So what else you can do with CRISPR technology? Well, Cas9 is usually cutting the two strands. This is why we call this effect the double strand break. So these are illustrated here on your right with this X. Now we can have an E case, and the E case is only cutting one strand. And also we can mutate the two domains that are responsible for cutting each strand, and we can create a Cas9. And Cas9 is no longer cutting DNA. So what could be the utility of a Cas9? Well, Cas9 can be associated with an activating domain, and it can be used to target at the promoter region of a given gene that is silenced, and you can promote the reactivation of the gene. So you can awake the gene, and this has sometimes therapeutic consequences. And you can do the opposite, of course, because you can associate this Cas9 with a repressing domain, and you can inactivate, you can silence a gene that is wrongly, that is pathologically overexpressed, and this also can have therapeutic consequences. which were the limitations that CRISPR-Cas9 tools had over the first generation of these CRISPR-Cas9 tools. So basically, so far, I only mentioned the first generation of CRISPR-Cas9 tools. Well, the limitations were twofold. At first, you could also target sequences that are similar, because basically you are guiding the nuclease with an RNA that is targeting 20 nucleotides. But if you have a sequence that is very similar, 19 out of 20 or 18 out of 20 nucleotides somewhere else in the genome, you will be eventually promoting the cut and the inactivation of this other gene. And this is bad news. Well, at the beginning, this was very much worrisome, and there were many people worried about this, and there were so many papers published. Nowadays, 10 years later, I can tell you that this is not so much of a problem, because we can use bioinformatic designing tools that are selecting guides that are unique in the genome, and we can even use recombinantly generated Cas9 nucleases that are even more specific than the ones existing in nature. And this is reducing to what I would say it's the normal rate of mutation. So this is take-home message. This is not the main problem of the CRISPR-Cas9 tools of the first generation. The real problem is the on-target effects. What happens at the place where you are cutting the DNA? Because this is triggering mosaics, and this is something we cannot control. Because I told you that the HDR, the right pathway, we can forget about. So it's not the preferred repairing pathway, no, we can control. So the non-homologous enjoining is progressing by randomly, this is the word, inserting and deleting. So basically, the message is that every time you have to repair the DNA molecule, you will be repairing the molecule in a different manner. And this is problematic because you will be generating dozens of alleles, and perhaps one of them will be the one that you are looking for. How can you visualize these mosaics? Well, since we are working with pigmentary genes, we can try to reconstitute the tyrosinase mutated albino mouse, and with a guide and with the corresponding oligonucleotide for correcting the tyrosinase. And we hope to get a mouse that is uniformly pigmented. Instead of this, we're getting this mosaic mouse. And this mosaic mouse, it tells you that only parts of the body, they manage to correct correctly, they correct appropriately, adequately the gene. And this is happening because we are injecting these CRISPR reagents at the level of one cell embryo. But the embryos dividing 2, 4, 8, 16, 32, etc. And the CRISPR reagents, they are not vanishing, they are not disappearing. They're still active for many applications. So at the level of an 8-cell embryo, because each blastome has 2 copies of a gene, you have 8 times 2, 16 possible alleles that can be corrected in different ways. And this is the problem. If you are not working with pigmentary genes, how do you see this effect? Well, basically you sequence, you use next generation sequence, you use deep sequencing, and basically you will be witnessing the highest variety of restoring, of repairing. So you have each one of these lines is a different mouse. So you see how many different mice we have been able to create. One of them is the one we were looking for, but the rest, they are not interesting. So this is something that we can do perfectly well in mice. Basically, we know that all the mice that are born after a CRISPR experiment, they will be mosaic. We have to identify the one that is carrying the allele that we're looking for, and we have to breed 1 or 2 generations and hope that these mutations will be segregated if they are traveling in different chromosomes, and eventually you will get a mouse that only carries your mutation of interest. But this is something you can do with mice, but you cannot do with patients. You cannot attempt to use CRISPR-Cas9 for modifying a sequence of this first generation tool. You cannot treat a patient and tell this patient, we don't know about you, but maybe one of your children will be inheriting the right allele. So this doesn't work like this. At the clinical level, basically, when we're treating a patient, we hope that the treatment will benefit the patient, not the children of the patient. And this is basically, this is the Achilles heel. This is basically the most important limitation. Sometimes this limitation is turning into an advantage, and this was nicely demonstrated by Paula Rio from the CMAT here and the CBRR. This is a colleague at the Rare Disease Institution. They are using as a rare disease Fanconi anemia, and as you know, the Fanconi anemia proteins, there are more than 20. They are involved in the proliferation of the hematopoietic lineages. So even though they are using non-homologous adjoining, because once they are restoring correctly the mutated protein, they are recapitulating and they are restoring the proliferation capacity of the cell, even though at the very beginning it might be one or very few cells that have only corrected the gene, corrected adequately the genes, these cells will be proliferating more than the rest, and at the end of the day will be taking over. So because of this proliferative advantage, these mosaics sometimes can also have a therapeutic side. So how do we solve the mosaics of the first generation CRISPR-Cas9 genome editing tools? Well, this is where David Liu comes into place. David Liu from the Broad Institute, they invented the base editors. And this is something that the nature, evolution has never invented. So the aminase has never gone in collaboration or in cooperation with dead-Cas9 or nickase. And what is the role of a deaminase? Well, there are different deaminases that can change a C into a D or an A into a G. And there is a third flavor of deaminase that is changing a C into a G. And this was originally described in 2016. And I want to highlight this. 2016, just 7 years ago. And as you will see, this is the tools. This is what we call the second generation CRISPR tools. These are the ones that are reaching the clinic. So in order to understand how this is working, basically the dead-Cas9 that is being brought into a given position in the genome by your guide RNA has this deaminase that is hanging, and this deaminase, you have to calculate the atomic size of this protein, and you have to calculate the guide in order that the active center of the deaminase is touching the nucleotide that you want to change. And of course, this is very difficult. And the first generations, the first editions of these base editors were very complicated. Now there are more than 10 versions of these base editors, and the last versions, they are really specific. At the very beginning, as you can see, well, very beginning, I'm talking 3 years ago, and you will see that, for instance, in this animal model of tyrosinemia, hereditary tyrosinemia type 1, the gene that was corrected, and as you will see in the bottom right part, you will see that this A in position 9 was changed into a G, but also the A in position 7, but also the A in position 5 and A in position 3, which means it's not so good news, because we're changing what we want, but we're also changing things that we don't want. So this has been already improved, and now pretty much the nucleotides that are changed are the ones that are associated according to your guide. So this is another successful example, and this is from Gerald Schwang in Switzerland, and this is a mouse model of a common rare disease. This is PKU or phenylcetonuria, in which this phenylalanine hydroxylase, the enzyme that is converting phenylalanine in tyrosinase, is missing, and this has two consequences. First, tyrosinase tyrosine is not produced, and since tyrosinase is used for melanin production, the PKU mice, they are lightly pigmented, as you can see, the one in the middle. And the other consequences is that the phenylalanine grows and accumulates, and this is neurotoxic, and this is really generating problems and mental disorders in humans if they are not, if you're not changing the diet of these children. You can use base editors, and the first mouse of these three is a base edited mouse in which the mutation has been corrected, And as you can see, that the pigmentation is also restored. The same has been applied for retinal degeneration in this animal model of retinitis pigmentosa, in which you can restore. And the same has also been applied to a mouse model of progeria. This is the accelerated aging. This is the Hutchinson gene for progeria. And this is acting on the lamin A gene. And this is a very unique and single mutation, C2T, that you can use base editors to correct. And with these mice, you can extend their life expectancy from the 200 days to more than 500, which would be as any other laboratory mouse. So this is already working. The last example from this year is also in mouse models. It's for a severe combined immunodeficiency in which they are using base editing for correcting the CD3-TD gene. This is also appearing in pediatric immunodeficiency. So as you can see, this is already working. Two more examples of base editing. So this is a mouse model for sickle cell disease being also a rare, but also rather common, especially in Africa and in Asia disease. And as you know, the beta globin is mutated. In this case, the mutation is not properly corrected, but is converted. This valine, which is a mutated valine, is corrected or is converted into an alanine. And this alanine is not the correct amino acid that should be there, but the alanine is already existing in the human population. It's called the Makassar allele. So as you can see, you can change this A into a G, and this will be no longer coding for valine, which is the mutated beta globin, but to an alanine. And the beta globin having an alanine at this position is not 100% active, but is partially active. And there are people living with this beta globin, alternative beta globin. And this is a therapeutic solution that can be brought into the clinic. And this is already in clinical trials. And the other example that you can think about base editing is not only useful for changing an amino acid one by another, but also for creating stops, for creating premature stop codons so that you can truncate a protein and generate a knockout. For instance, you have a CAG that encodes normally glutamic acid. And if you change the C into a T with the corresponding base editor, you will be generating a TAG, which the TAG converted into UAG into RNA, which is a stop codon. And why this could be useful? Well, this is the work done by Kiran Musunuru, in which they have been using base editing for inactivating PCSK9. And PCSK9 is the repressor of this LDL receptor. This is these low-density lipoproteins. And these low-density lipoproteins are the ones that are carrying the cholesterol. If we are inactivating the repressor of the receptor is increasing in the hepatic cells. And if the receptor is increasing, it's also capturing, it's actually importing more LDLs from the bloodstream. So the final result is that the circulating LDLs, they are lower and the cholesterol level is lower. So this is associated with a reduced risk for atherosclerosis and reduced risk for cardiovascular diseases. This is how it's been tested in macaques, in which a single administration of these base editors is resulting not only in a reduction of the PCSK9 as expected, but also a reduction in the LDL circulating levels. So these are the two companies, as I mentioned before, Beam Therapeutics for the sickle cell disease clinical trial and VHIR Therapeutics for the PCSK9, that already last year, they have already started to test this into patients. So this is already in the clinic at the level of clinical trials. So this is why May last year, this editorial in Nature Biotechnology was confirming that not the first generation tools, but the second generation CRISPR tools would be the one that will be eventually reaching the clinic. Well, David Liu was not only producing the second generation tool, but also the third generation tool. The third generation CRISPR tool is the prime editor. Also playing again with the meaning of the word. Prime means not only priming an oligonucleotide, but also prime means quality. No? We all know a courier company that if we are purchasing our goods through the prime system, they arrive next day. So everybody understands that prime editing means better editing. And this is exactly what they wanted. In this case, David Liu, they associated reverse transcriptase to an E-case. And this is another crazy idea that evolution never thought about this. So there is no bacteria, there is no animal or plant or yeast on Earth that has ever combined a reverse transcriptase with an E-case. This is only coming from the imagination, from the talent of David Liu. And what is the point about using reverse transcriptase? Well, this is a graphic that is illustrating how it works. So the guide RNA is no longer called guide RNA, it's called PEC RNA, and it has a 3' end that is very long. It's so long that it also can be complementary to the complementary DNA strand. I know this is difficult to visualize, but basically we're using still the guide at the 5', the RNA guide, for target our gene of interest, but the 3' of the RNA is targeting the complementary DNA. And also this coincides with the active center of the reverse transcription, which is producing DNA from RNA. And now we can dictate what is the DNA sequence that we will be producing, because we are dictating this with the corresponding sequence at the 5' end of this RNA. This is fascinating. This is one of the most evolved tools. And what is the consequences of using prime editing? Well, this is one of the first examples of comparing back-to-back first generation tools and third generation tools in mice. So on the left, this is first generation tools. This is classical CRISPR-Cas9. On your right, this is prime editing. As you can see, the efficiency of on targeting is the same order level. So it's 56% to 21%. So it's similar order of magnitude. But what is the main difference is that the level of indels, the levels of mosaics is, as usual, for the CRISPR-Cas9 is very high, 40%, but for the prime editors is almost non-existent. And this is the path for our success. Prime editors have not yet reached the clinic. They are expected to be reaching the clinic in 2024, but as you can see, they have a good future ahead because they are already correcting one of the main problems of the CRISPR technology, such as these mosaics. Similar to what happens to base editors, with prime editors, we don't have one single version. We have more than 10 versions. And if you go to Agin or any other repository, my recommendation is always to select the latest version, because there are many improvements that you can apply in order to use the latest tool that is available. So this is a revision from David Liu, one of the last revisions from David Liu, which is basically, this is where we are today with the CRISPR technology. We can use nuclease editing, we can use cytosine-based editing, we can use adenine-based editing, and we can use prime editing. These are the graphics on the left. On the right, there are the different applications, whether you want a small insertion or small deletion or a large insertion or large deletion, and then it tells you what are the most recommended tools that you have to use. So eventually, we want this to be used for therapy. And we know that we had a bad start with the therapy, because we had, in November 2018, we had this guy, He Jiankui, in Shenzhen, that pretended to use the CRISPR-Cas9 first generation to inactivate CCR5, which is the core receptor that is being used by HIV that is causing AIDS. He wanted to create a subset of human beings that were resistant to HIV. Well, he failed. He managed to create 3 girls, 2 twins and a third girl that had all the different problems that we have seen in mice. So he not only did not create the intended mutation, but also he was able to create additional unwanted mutations elsewhere in the genes of these poor girls. And also he was not controlling the experiment. And this was bringing him to jail, and he was being liberated last spring, not this spring, but 2022 spring. And this is something that belongs to the past, but at least has to be mentioned, because this is an example that not always researchers, they do the right thing to do. So this was an experiment that had not to be done, but eventually was done, and now we have to learn from the experience. What the gene therapy we are focused, the one that is legal, the one that is authorized in many countries, is the so-called somatic gene therapy. We know this because we have 2 different types of it, the in vivo and ex vivo. In vivo, very easy. We are delivering directly the CRISPR tools into the patient. We're hoping that these CRISPR tools will be correcting or editing, will be doing something that will be therapeutically interesting. The ex vivo, it's safer because we are extracting cells from the patient. We are editing the cells when we are growing them in the laboratory, and the edited selected cells are the ones that are returned to the patient. So this is safer because we are selecting what we are returning to the patient. However, as you can foresee, we cannot apply ex vivo to all different pathologies. We can only apply this only where we have cells that are available, cells that can be used for replenishing organs or replenishing systems. And I'm basically referring to the blood cells or to the skin cells. We cannot apply this for muscle cells. We cannot apply ex vivo for neuronal cells. The first of such experiments were online presented at the end of 2015, so almost only 2 years after we started using CRISPR first generation tools, and eventually they were published at the beginning of 2016. And the 3 labs independently, they used the same animal model, the MDX mouse. MDX is a model for Duchenne dystrophy. It's not exactly a model for Duchenne dystrophy, but is a model for the dystrophin mutation. The dystrophin is really severe, and it's causing the death, unfortunately, of the people having Duchenne dystrophy in the 30s or sometimes in the 40s. But here we can mimic, and we can try to recover one of the most common mutations that are affecting one of these exons. This is the 23 exons. Mind you that the dystrophy gene is one of the biggest genes we have in the mammalian genome. It's 1.2 million base pairs, the dystrophin gene. So we can target left and right to this exon, remove this pathological exon, and create a resulting dystrophin that is reduced size, that has not this exon, but that is partially active. And this is what you can see in the right image, in which maybe 10–50% of the muscle fibers, they are recovering the function of the dystrophy. So people thought that this would be interesting to test not only in mice, but also in dogs. And these are beagle dogs in which 2 years later, they were demonstrated that it is possible. But I draw to your attention to the efficiency of the process. And the efficiency of the process goes between 3 to 92%, which is the same as to say between 0 and 100%, which means in real terms, sometimes it works, sometimes it doesn't work. So this is not ready for jumping into the clinic, as you can see. Nonetheless, even though it wasn't ready, there was a company, Cure Rare Disease in Boston, that they decided to treat this person. This is Terry Hogan, and this is the brother of the CEO of this company. And they wanted to use one of these activating DEDCAS9 systems, one of these epigenetic use of CRISPR, in which they were using the PP64, which is triggering the activation with a cryptic promoter of the dystrophin gene, because this guy had a mutation in the first exon. Well, this is an example of N=1 therapies. So it's just one patient being treated. I'm really very much against these attempts. So I know there are people that's very fan of this, but with an N=1, if everything goes well, it's fantastic. But if this doesn't work, you know nothing, because there is no controls. In this case, the problem was that you can treat mice. The mice, they weigh 25, 30 grams, but this person was weighing about 70 kilos. And when you do the maths and you calculate the number of the viral vehicle that is delivering these CRISPR tools, these adeno-associated viral, you have to use 10 to the 14 per kilo, which is for 70 kilos, it means that 10 to the 15. So you are really dumping, you are really saturating the body of this person with adeno-associated viral proteins. What happens is that 1 week after the treatment, he died out of an immune reaction. So he was mounting a cytokine storm against all these different adeno-associated viral particles, which tells us that it is not as simple as multiplying what is working in mice, what should be or could be working in humans. So we have to be careful in transferring the experience from animal models into human beings, particularly when we're using these adeno-associated viral proteins. This is not the first case in which treatment with higher doses of viruses, because you have to reach all the muscles in the body and you have to give them through the bloodstream. And this is why you have to use such a high dose, but these are risky and this is dangerous. So the gene therapy at the very beginning with CRISPR, as I was telling you at the beginning of the seminar, we all thought that would be applied through the homology repair, driving repair, in which we will be cutting next to the mutation and we will be providing an oligonucleotide, and eventually this will be corrected. Just give me a second. Okay, because there was some noise in the lab. Basically, so this is the gene therapy that was prepared or that was imagined at the very beginning. In reality, this was not working. What is currently being used, if we cannot modify a gene, we can use CRISPR for what is worth. And what is worth is using CRISPR for inactivating alleles, because CRISPR is very good at it. Remember, biologically, the CRISPR is used by bacteria to inactivate these alleles. So basically, if we cannot modify the incorrect allele, what we can do is to correct and to target and mutate this allele. And this is what is being done. So there are about 90 clinical trials at the moment using different types of CRISPR. This is a few hundred, there's a couple of hundred patients that are already undergoing CRISPR treatment. So this is already occurring. So there is no single CRISPR treatment that has been approved. All what you hear, all what you see in publications are the results of phase 1 or phase 2, and in some cases, phase 3 clinical trials. Most of them are ex vivo, and most of them are applied for the cancer immunotherapy. These are examples of different use of Cas9 or base editors in different type of disease. I'll give you some examples. For instance, CRISPR in cancer immunotherapy. Well, you know, the CAR T-cell treatment. You know that in 2018, James Allison and Toshiko Hanho were receiving the Nobel Prize in Medicine and Physiology because they discovered the PD-1 and the CTL4 immune genes. These are the negative regulators. Well, these are the genes that are normally inactivated in this CAR T treatment, because when you have people with melanoma, sarcoma, or myeloma that can no longer be treated medically, you can extract T cells from them, and you can apply CRISPR to inactivate the T cell receptor and to inactivate the PD-1. And before returning the T cells into the body, you can transfer with a lentivirus this chimeric antigen receptor that is directed against these melanoma cells, and this will be aggressively treating and aggressively attacking these cancer cells. Well, this unfortunately doesn't work with all patients, but it works with a respectable, with a significant amount of patients, and this is probably the most frequent use of CRISPR in the clinic nowadays. This is the example I was referring at the very beginning of the seminar. This is Alisa. In May 2022, was treated of their acute lymphoblastic leukemia, and they were using base editors, not CRISPR-Cas9 first generation, but second generation, to inactivate the T-cell receptor, the CD7 and the CD52. Why these are inactivated? Because these are the targets of the chemotherapy drugs. So you are preparing a CAR T cell that has the chimeric antigen receptor, but has inactivated the receptors that are transducing the chemotherapeutic agents. So these are the lymphocytes that are surviving the chemotherapy, and other ones that are replenishing, and they can also restore the hematological system of this person. This was eventually published, and there were 3 kids treated. 2 of them, they were successful, and 1 of them was not successful, but not related with the base editing. It was related with an infection that was occurring to this 3rd child, because of course these are severe immunodeficient kids. Well, another success that has to be mentioned, and this is what I was referring before when I said Emmanuel Charpentier is the most successful. As you can see, CRISPR Therapeutics is here together with Vertex. Basically, they developed this treatment that was envisaged many years ago by Stuart Orkin at the Children's Hospital in Boston. Basically, is to treat sickle cell anemia with a very talented idea. So the gamma globin that is working as the beta globin during the fetal development, the gamma gene is switched off at birth, and the beta globin is taken over. But the gamma globin is switched off because there is a repressor called BCL11A that is sitting on the top of the promoter and is preventing its transcription. If we inactivate or we reduce the levels of the repressor, then we allow the gamma globin to be expressed in the adulthood. And this is already happening naturally in some people, and this is why they realize about this possible therapy. And this is actually what has been done, and eventually the gamma is replacing the beta globin, and this is Victoria Gray. This is the first person in the world that was treated with this CRISPR. This is first generation CRISPR-Cas9, because basically what we are treating, what we are inactivating is the enhancer of the BCL11A. We cannot inactivate the gene itself because the gene is required for other functions in the hematological system. So Victoria Gray was treated in summer 2029, and at the end of 2020, beginning of 2021, it was appearing in New England Journal of Medicine. And as you can see, the evolution of the first 1.5 years, the green bars, which represent beta globin, were replaced by the blue bars. And at the end of last year, about 50% of the patients that were treated this way were successfully cured. So this is a single administration, and these people are cured for life. Well, for life, since they have been treated. We just hope that this will be maintained for the following years. I met Victoria Gray this March because I was attending this 3rd International Summit on Human Genome Editing that was taking place in London. This is the 3rd summit after the first one in Washington and the second one in Hong Kong. And Victoria Gray was so thankful and grateful to these people, because of course she knew that she was the first person being cured. But she had a sad face, and the sad face because she realized that not many people would eventually benefit. This is, as I said, this would be the first treatment that is likely to be approved this year, 2023, by EMA and by the FDA, by the European and the US regulatory agencies. The problem being that the cost of this treatment will be around 3 million euros per patient. And this is the problem. So how can you envisage the 300,000 cases of sickle cell anemia that are year after year appearing in Africa? How can you imagine that all these people will be receiving this at 3 million euros? So basically, you have to compromise the very legitimate way that the pharma companies have to recover their investment, but also with a rational price, with a price that is payable not only by individual people, but also by the national health system. So the reality is that basically we are left with a problem in which scientifically and therapeutically speaking, we have corrected a disease, and now the limitation is no longer scientific or therapeutic, but the limitation is economic. So there are other cases in which the technique did not go so well. This is for treatment of retinal degeneration disease. This is Leber congenital amaurosis type 10. This is a rare disease, and this is to eliminate a cryptic bronze sequence in one of the introns that is triggering an incorrect processing of the gene. And this has been already in the clinical trials, and only out of the several dozens of patients being treated, only 2 or 3 they are reporting an improved vision. This is one of the persons, Carlene, that has been reporting this improved vision. This is treatment in just one eye. This is a monocular, this is one eye that is being treated. Another example that I will have to share is to tell you that so far, all the different clinical trials are using adeno-associated viruses. And you know that if you're using them for eyes, the dose and the amount of viruses you have to use is very much reduced. And if you're using them for treating muscle cells, and as you saw the danger, so you are risking the life of the patient you are treating. So what are the alternatives? Well, the alternatives are the one from Intellia. And Intellia, you might remember, that's the company from Jennifer Doudna. And Intellia demonstrated in 2018 in a mouse model that they could treat in a single dose administration a rare disease. It's called transthyretin amyloidosis congenital, with the gene TTR being overexpressed. So the treatment is to kill the gene, to inactivate the gene, which is targeting neuronal and muscle cells, and eventually is resulting in the death of the patients. So basically, they chose not to use adeno-associated, they chose to use nanoparticles, actually lipid nanoparticles, to encapsulate 2 RNAs, the one encoding the Cas9 and the one encoding the guide RNA. Eventually, in summer 2021, they validate this in, this is the particles they use, and as you can see, the 2 RNAs. They also opsonize these particles with a polypropylene E so that they can be better captured by the liver. This is where this gene is being expressed. And also, when they tested this in macaques, in non-human primates at different doses, you see that after 1 year, after one single treatment, the level of the TTR gene expression was maintained low. So this is already into the clinic. This is already in clinical trials, and this is Patty Doherty. And this is one of the first patients where it has they confirmed that the strategy worked. So again, this is illustrating that this is already reaching the clinic at the level of phase 1 and phase 2 clinical trials, in this case with reducing the expression of the gene. So these lipid nanoparticles, it might be familiar to you because these are the same lipid nanoparticles that are encapsulating the RNA of the spike protein of the coronaviruses, which is the 2 of the most famous and most administered vaccines in the worldwide, which are the ones by Pfizer, BioNTech, and by Moderna. So as a summary of this seminar regarding therapy, where we are at the moment, it's more than 200 patients being treated. Actually, it's more than 300 at the moment already. If we want to inactivate genes, what we can use is either CRISPR-Cas9 first generation or base editors second generation. If we want to correct genes, it means changing the sequence, single base substitution. We can use base editors, and maybe in the future we could use prime editors. Prime editors, they're also good for insertions, small insertion and small deletions. And the technologies that we will use for the deliver, that would still include and being reduced the adeno-associated viruses, also the lipid and nanoparticles. This is an example I took from before the summer, and in which just illustrate that there were at the time, and they're still there, 4 different clinical trials for the same disease, sickle cell disease, that they are using different approach, they are using different versions of the CRISPR-Cas technology. This is to illustrate how powerful and how rapidly this is progressing. Let me finish off in these minutes that are left with a development that we published at the beginning of the year in 2 papers. Because so far, what I've been referring, either if I was commenting on the first, second, or third generation, they're all coming from the standard Cas9. And the standard Cas9, they're coming from the very well-known bacteria, Streptococcus pyogenes and Staphylococcus aureus. Pyogenes is causing otitis and laryngitis, so it's causing human infection very often. And aureus, what can I say of aureus? Staphylo aureus is present in all hospitals, unfortunately, and is together with some fungi infections. Staphylo aureus is responsible for most of the infections that are appearing in immunodeprived patients in the hospitals. So what it means, the bottom line, it means is that most of us, we have been exposed to these bacteria and to all their compounds, including the CRISPR-Cas9 systems. And this is something obvious, but it wasn't obvious to everyone until Matt Porteous, he realized and eventually published this in Nature Medicine in 2019. And he confirmed that most of us, we're carrying antibodies against the aureus and against pyogenes, which means we could not use this for therapy because we will be causing the immune system to react against the therapeutic tools. So before the therapeutic tools would be able to act, they will be removed from the blood circulation. So we are left with 2 solutions. Either we are applying immunosuppression together with the gene therapy, which is not very good news, or we go to find alternative Cas proteins. There are many CRISPR-Cas systems. So this is one of the last classifications of the CRISPR-Cas systems. There are different classes, there are different types, and there are different combinations. The easiest is the type 2, which are the ones that are using only one effector protein, Cas9, and one guide RNA. This is why we're using this type, but there are many other CRISPR-Cas types. And we don't know whether some of them will be even more efficient and even more successful for the therapy. Particularly, we want to use CRISPR-Cas systems from bacteria that had no relationship with the human beings. And here you can do 2 things. You can travel in space, so you can go to places, remote places on Earth that have not been colonized by human beings. This is the Atacama Desert in Chile, or the Antarctic, or the depth of the Marianas at the ocean, or you go to these basins in the Yellowstone. These are very strange places where there are bacteria that never ever had any contact with the human beings. And this is something successful that has been done. But of course, you need money. You need money for all these expeditions, and you need money for this. When we don't have money, but we have still good ideas, what you can do is you go to the lagoon that is next to where you live. And this is what Francis and we have done. Francis suggested to go to El Hondo. El Hondo is a lagoon next to Elche, and Francis, mind you, he lives in Elche. And there, in the water of this lagoon, we managed to obtain a new CRISPR-Cas system. This is a smaller Cas9, EH means El Hondo, and we characterize, and this is where the El Hondo fits into this genealogy tree of CRISPR-Cas system, and this is it's about 8,000 amino acids. The standard Cas9 is about 1,400 amino acids, so this is smaller. And the question is, how does it look? Well, nowadays with the AlphaFold features, we can anticipate the structure of this Cas9. It's very similar to the Cas9 that we know already from Biogenes. And in combination with a company, Cas9, we have managed to uncover the PAM sequence, which is not as simple as the one from Biogenes. So you see here in red, this PAM sequence. And the most important, we tested this in human cells and mouse and human cells, and this was effective, this was efficient. So, in particular, this was efficient in a gene whose target was next to a sequence that was exactly compatible with the PAM. Thereby, we confirm that it is important for this new Cas9 to have this particularly PAM. So this is an example that you can find Cas9 nucleases somewhere else. So, and this is what many people are doing. But we wanted to go beyond. So we wanted to not only travel in space, but travel in time. So if you know the movie Back into the Future, so basically we wanted to do a Back into the Future sort of thing, because we envisage what would be the CRISPR-Cas systems that would be operating in bacteria that could have lived millions of years ago. The problem with this approach is that the oldest DNA that we can have, it's about 2 million years. So this is already killing the script and the argument of Jurassic Park. So the dinosaurs, they were extinct in 65 million years ago. So there is no DNA surviving from 65 million years ago. The oldest DNA that we have been able to rescue, it's this 2 million. Remember that not this year, but last year, Nobel Prize Svante Pääbo is using paleoenzyme, paleoDNA, so it's moving between 10,000 to 400,000 years ago, which is already a lot, but it's not what we wanted. So what we decided is to go really back in the history of life. So this is sort of a summary of the history of our planet. So our planet was formed about 4,500 million years ago. 1,000 million years later, the first prokaryotic cells were appearing. Around 1,850 million years ago, the first eukaryotic cells appear. Around 600 million years ago, there was the Cambric explosion. Many of the existing species, they were derived from the original beings that were created at this time. And of course, 65 years ago, the dinosaurs were extinct. So we decided to create a CRISPR-Cas system from 3,737, 200,000, and 2,600 million years ago. So you can ask, if there is no DNA older than 2 million years ago, how did you manage to produce this? Well, we managed because we set up a collaboration with Raúl Pérez Jiménez. He's a paleoenzymologist, and he's able to resurrect this ancestral Cas9. And for doing this, we apply this maximum likelihood method. This is a reconstruction technique in which we are providing to a supercomputer with the sequences of all the CRISPR-Cas systems from many bacteria that are evolutionary related, and we are telling the computer to look for an ancestor that could explain the diversity of Cas9 existing nowadays. And when the computer finds this protein, we tell the computer to find the ancestor of that ancestor, and the ancestor of the ancestor of the ancestor. And we can go millions of generations back in time with a chronological order. And so we can go to sequences that we don't know exactly whether they existed or not, but are most likely the proteins that could have existed that explain the current nowadays diversity of Cas9. So the proteins that we were able to obtain from the computer, they have identities between 50 to 96%. To our surprise, also using AlphaFold, there were highly similar structurally to the Cas9. So even though the sequence, the primary sequence is very different, when we compare the structures, they're also very similar. Regarding activities, when we go back in time, the oldest, they have nicking activity, whereas the newest, they sort of lost this nicking activity. Nicking, it means cutting one strand of DNA, and they have recovered the double strand activity. The oldest, they seem to cut single strand DNA better than double strand, and they seem to cut RNA. And the newest, they are cutting not only this, but also double strand DNA. And this also makes sense, because millions of years ago, at the beginning of life, I mean, the most plausible explanation is that before DNA, we were living in an RNA world, in a single strand world. And it is likely to expect that the bacteriophages that were infecting the first prokaryotic organisms, they were also single strand RNA. So you would expect that the nuclease would be also cutting single strand. And this is exactly what we found out. And most importantly, in this color graphic at bottom right, so you see the reactions to the antibodies that normally are present in the human blood against the Cas9 from Pyogenes. So in red is the reference, the Pyogenes, and in the other colors are the three examples of this ancestor in which you see there is almost no reaction. So we're fulfilling the hypothesis. We are obtaining Cas9s that are not cross-reacting with our antibodies. And the question is whether they are working. Well, first, whether they need a pump. The older we go, the less pump they require. And this also makes sense. At the beginning of life, there was less complexity. There was probably less number of viruses, less number of bacteria available, and it was not so relevant to discriminate between the virus and the endogenous genome. Whereas as we progress, the newest being 37 or 137, you need to use a particular pump. And this is why you see that the first two bars in blue and red and green, which means the oldest ancestral nucleases, are the ones that they don't care and they work no matter what pump. Whereas the newest pumps, they need particularly pump activity. The most important graphic of this collaboration was whether they work in human cells. And this is the results in using two genes. In red is the reference. It's normalized to 100% to Cas9. And when we go 37 million years ago, you can see in purple, 60% of the activity is still retained. When we go 137 million years ago, you can go up to more than 80% of activity. This is amazing. And when we go 200 million years ago in yellow, it's about 40%, the same for the 1,000 million years ago in green. The only one that we could not validate this operation was the oldest, which is 2,600 million years ago, which is also logical because this was a nuclease that was selected to operate against single-strand molecules, not double-strand molecules. Last but not least, this we eventually published at the beginning of the year, and in collaboration with Miguel Ángel Moreno Mateos in Sevilla, we demonstrated that this is not only working in cells, but also is working in zebrafish. So it's working already in vivo in zebrafish, which means that we have been able to obtain these new nucleases that now can eventually be tested for therapeutical applications. As a matter of fact, there is a company that it's already testing this or will be testing this in clinical trials. Well, I think we have 10 minutes left. I just want to thank my collaborators for this last experiment. This is Francis and Raul. And let me tell you that this is an experiment that was never funded. So no one believed we were capable of doing this experiment. We were pre-selected in different goals, in different foundations and different agencies. but we failed at the interview level because everyone in the interview, they were asking us for an example of a resurrection before funding us. And we always said, if we have an example of a resurrection, we no longer need the project. We already demonstrated the project. So you want a risky project? Here's a risky project. And of course, eventually we decided to go to take over ourselves the funding of this project, and we were successful. So let me thank the people in the lab and the different agencies and associations and foundations that are providing us funds. And also let me thank the Association of Helping People with Albinism that we are collaborating with. And this is my last slide. And now we have 10 minutes for any questions that you might have. Thank you.
Okay, Luis, thank you very much for this outstanding and inspiring talk. I think, well, this has been a great example of how science is moving in our country and
Well,
everywhere.
well, I mean, the people participating, they always get the latest results, but you can compare from year to year that the things are changing. I'm never repeating this talk. This talk is always different.
Yeah, this is what I have realized, and I was, yes, the talk is improving and all the advances that are coming. So, well, so that the talk is open for discussions. Yeah. So if there is any question, you can open your microphone or write it in preguntas y respuestas. We have it here down in the screen.
Or the chat, or they could use the chat as well.
The chat, yes, there is one question.
Okay, there is one question. Lola Medina is asking me, will CRISPR be able to reverse aging? Well, if we knew the genes that are associated with aging, that I don't think we know, if we could target these genes, you could envisage that you can not reverse, but you can reduce, or you can extend the life expectancy. And this has been done with some animal models in C. elegans and in some cases in mice. But I'm a bit doubting that this will have a therapeutic effect or therapeutic meaning for human beings, particularly because we know nothing about, or we know very little about the genes that we will have to target. More questions?
Okay. Well, it was in the meanwhile, how do you visualize the close future for the clinical use? Because it has been funny because all the questions that were coming to my mind during your talk, and you were answering all of them. So, but now you have said we are in the present. How do you see the future, the close future for clinical use? I mean,
well, the close future would be that this becomes a reality, because the few hundred people that have been already treated with CRISPR tools, they have been treated at the level of different phases of clinical trials. What we need now, we need a proper set of drugs or medicaments or gene therapy medicaments approved, and we need a long cohort of people, a large cohort of people that are treated, because we have to investigate the long-term effects of this, because we don't know about this. And the reality is that in 2023, the first CRISPR applications will likely be approved if everything goes well. In 2024, we will get the first prime editors reaching the clinic. And I have a lot of expectations with the prime editors. People is a bit afraid about overexpressing a reverse transcriptase in human cells, but with the latest versions of the prime editors, and I think they're safer, and I think they will be revolutionizing. They will be boosting again at the same level they already boost the base editors. So we have Craig Vincent. Thank you. Thank you. Do you know of any clinics using? Yes, the DEDCAS9. Well, unfortunately, the clinical trial was not a clinical trial. It was a compassionate use. It's the Terry Horgan case. It's the Duchenne guy, and he was 27 years old and he was treated with VHIR associated with PP64. And the idea was to reactivate a cryptic promoter of the dystrophy gene, because the mutation was affecting the first exon, and it was kind of skipping the first exon with the mutation and reactivating the dystrophin downstream of the first exon with a cryptic promoter. The problem is that, as you can see, the delivery was dangerous. The delivery, the millions of adeno-associated particles were the ones probably killing this person. Actually, there is a publication that you can check in which they investigated why this person died. And this person actually died not only because of this, but also because they didn't know this at the very beginning. He was more susceptible for generating this cytokine storm, so which was a bit unfortunate. So this guy was not only affected by the Duchenne, but also was affected by a kind of a disorder that was creating these altered responses.
Luis, one question. Do you think? Yes.
Let me finish the second part of Craig.
Okay.
Whether this technology has any clinical advantage over Cas9 editing. Well, properly used, if you can reactivate the gene, it will be the solution for many pathologies. For instance, if you think about the X chromosome fragile syndrome, or if you think about the Rett syndrome, in which these are syndromes in which the genes that are involved are repressed. And one way of mounting a therapy for these pathologies which are severe is to try to reactivate, to recover, to awake these genes. This hasn't been tested yet in clinical trials, but I envisage that this could be a solution, particularly for those diseases in which no gene therapies are available.
Another one, Andreina. Yeah.
Is there any study being conducted to modify embryonic genes to prevent phenotypic expression? Well, I mean, this has been done in mice. I mean, there are many studies, and in mice you can find many studies. And for instance, you probably remember beginning of the summer in June, there was this discussion. It was a lot of controversy by Jacob Hanna and Magdalena Sernica-Goetz that they were producing these synthetic embryos. First, synthetic mouse embryos, and second, synthetic human embryos. And in some of these protocols, they were using CRISPR to inactivate certain genes in order that these embryonic cells spontaneously, they will be reorganizing as embryos. But this is something that is in its infancy. So this has to be moving. So Paula, are all the clinical trials based on Cas9? No, absolutely no. There are several trials that are using Cas12, and there are others using another version of Cas. So this is being open. And actually, this was one of the reasons of the two examples that we published this year, in which we characterize a Cas9 from a lagoon, from another place. It's a different Cas9, so it's not the standard Cas9, and also this ancestral Cas9. So even though we call them Cas9 because of the scheme and because of the domains they have, they are different from the pyogenes and different from the Aureus. And definitely 12 is involved in some. If you see, when you will get the slides, there's a couple of examples using Cas12.
I think, Luis, do you think that the use of different nanoparticles will help to reduce this anti-Cas9 response that we have, that many people has?
Well, I mean, these lipid nanoparticles is the future. And this is the largest experiment ever done. There are 11,000 million of doses of vaccines, RNA-based, that have been administered to human beings across the whole world. So that's the largest experiment ever done. So it means lipid nanoparticles are safe. Lipid nanoparticles, they can be modified to target one or the other cell, because you can include in this surface of these particles peptides, or you can include proteins that are binding to particular receptors. So you will be directing these particles to a given cell type. So I guess what we will see is a reduction in the use of viral particles and an increase in the use of nanoparticles. Nanoparticles, we treat them as if they were only one type of nanoparticles. There are many. There are dozens and dozens of types of lipid nanoparticles. There are different combinations of lipids, different combinations of substance, and there is a lot of competition going on. Good competition, it means that we are developing newer nanoparticles. We are collaborating with some teams here in Spain that are developing nanoparticles, and I have to say I'm amazed how many different lipid nanoparticles they can produce by simply combining different lipids and different molecules.
Okay, Luis, the last question, Jimmy Vargas.
Okay, I'm going. So Jimmy,
yeah.
Is there a study related with the capacity or potential of the different types of Cas9, or mainly are all the same regardless of the microorganism or origin? Well, that's a good point. That's a good point. That's a good question. I'm not aware of such a study. There have been comparison between Cas9, so it means third generation and second generation and third generation. There have been comparison between Cas9s of different, for instance, Cas9 from Staphylo aureus is as functional as Cas9 from Pyogenes. The problem is that both, we have antibodies against both, and this is the problem. But basically, in this case, regardless of the microorganism, they seem to be working. But the comparison that you are suggesting, I think it would be interesting to be done, but I don't think it has been done properly. I have to look in the literature, but it will be anyhow an interesting experiment to do. So thank you. So it's 11. So
yeah, it's 11. Yes.
for everyone, and I hope this was useful. And at least the take-home message is that you have been getting the latest. So what we know today, that is 16th of October, about the CRISPR technology in biomedicine. And next year will be different.
Okay, so thank you very much, Luis. Thank you, everybody. And we're waiting for you next year to know the novelties. And so thank you, and see you.
Bye-bye, everyone. Bye. Have a good day.